All right, fantastic. So yes, hi, my name is Meghan Barrett. I'm an assistant professor at University Indianapolis, director of the Insect Welfare Research Society. A little bit about me. I'm an insect neuroscientist and physiologist by training, but I've actually done a decent amount of sampling of bugs using both active and passive methods for a wide variety of research questions in my past. So I've done bee balls, and I've done malaise traps, and I've done pitfall trapping. So for a bunch of different insects I was interested in at various points in time. And so today we're going to be talking about what we know and a lot about what we don't know about humane insect collection. So I just want to kind of give you the overall summary in case you have to go early. What we know, insect collection is more humane insofar as it follows the three R's, which we'll talk more about in a little bit, but are some principles of ethical treatment of animals in research settings. What we don't know, exactly which practices are best according to those three R's. And so the upshot of this basically means that a lot of what we're going to be talking about in this discussion today is that we need to be able to learn to live with uncertainty. We need to know that we need to keep searching for more information. This is a very new field, and we need to be really careful about what we commend and what we criticize because there's a little bit of a lack of information out there about a lot of these topics, and a lot of us are boxed in on particular methods for scientific reasons. So I'm going to start today by trying to motivate the question of why we care about humane insect collection at all. And so I'm going to talk a little bit about insect sentience, where sentience is often seen as the grounding for moral consideration for non-human animals. Then I'm going to talk about how we could ask new questions when we're collecting and best practices according to the three R's. After that, I'm going to go through a list of refinements for lethal methods. So I was sent a list by the folks at the Liverpool Museum of some common methods that are used for collecting in the UK. And so I'm going to go through what we know and don't know for each of those, as well as some anesthesia questions. And then finally, I'm going to end very rapidly with some resources and help. I'm going to blast through those slides because, of course, you can always come back and take a look at them later in the recording. Okay, so sentience is defined as the ability to feel positively or negatively balanced affective states, things like pleasure on the positive side or pain on the negative side. And by that, we mean more than nociception, which I'll go into in a minute. Now, many moral theories are going to suggest that we should consider the welfare of and avoid causing unnecessary pain in these beings that could be sentience candidates. So that means that we need to know which animals, which beings can experience pain. And therein, we're going to get to very quickly an empirical challenge, a puzzle. So nociception is the reflexive response of the nervous system to noxious stimulation. So imagine you reach your hand out and you touch a hot stove. The sensory cell in your fingertip is going to send a signal up to your spinal cord. Your spinal cord is going to send a signal back down to your motor neurons, and you're going to pull your hand away really, really fast. And that's a nociceptive response to that noxious, damaging stimulus of the hot stove. And that can happen without you feeling the pain at all. It can be a reflex. But at the same time, your spinal cord is sending a signal up to your brain proper, and there it's generating the feeling of pain. And so that's when you say, ouch, and you reach for the burn gel and you learn, presumably, not to touch hot stoves ever again. And so those kinds of responses are associated with the subjective experience of pain, right? And that subjective experience is more what we care about morally than that nociceptive reflex response. But of course, there's a fundamental challenge with subjective experience, which is that it's really hard to objectively see subjective experience. And so in humans, we can just ask other people what subjective experiences they're having. And assuming they're not lying to us, we're getting an honest report. And we call that data self-report data. So are you in pain? The person says, yes, I'm in pain. Great, we have evidence of pain in that individual. But unfortunately, we can't ask a lot of non-human animals to self-report whether they're having a subjective experience to us. And this has led in the past to some decisions that we might now think morally aren't as well grounded. So for example, many folks in the 17th century, actually all the way up through the 19th century, practiced vivisection in dogs. And the idea there was that the kind of kicking and the screaming of the dog was, just a mechanical reflex response to the noxious stimulation of being dissected alive. And nowadays we might think, actually, we're concerned that dog might have a subjective experience and not just be a little nose-offensive reflex machine, and we might want to give some analgesia prior to that dissection for that reason. So to avoid making the kinds of mistakes we might think we've made in the past with the dogs and the vivisection, we might want to look for objective proxy criteria that make it more or less plausible to us that a particular animal feels pain, even if we can't directly observe or access their subjective experience. So when we do that, we tend to look at two different kinds of information. We look at neurobiological information because we believe that to be kind of the physical substrate responsible for producing pain experience. And we look at behavioral evidence because we think that pain evolved in animals for a reason. And so if we look at their behavior and the kind of fitness relevant or adaptive benefits of these responses, we might get a better sense of which ones may be capable of experiencing pain. And so a lot of folks who are interested in this question of sentience in animals will take a framework-based approach where you kind of a priori establish neurobiological and behavioral criteria that you think are relevant to building the case for or against sentience in animals. And then you go and you apply that framework to different groups and see how many of those criteria they meet. So this is one framework. It's called the Birch Framework. It was created in 2021 by Jonathan Birch's group at the London School of Economics. He works on the Foundations of Animal Sentience project there, or he runs it, I should say. And it's based on a framework from like the 1990s by Smith and Boyd, 1991. And so they're very similar. And that framework was developed primarily for use in vertebrates. This framework was designed to help kind of increase the usability of the framework for invertebrates. So when we look at those 8 criteria, 4 of them are neurobiological, 4 of them are behavioral. And essentially the framework works by assessing, how strong the evidence is in each category for particular groups of animals. And the hope is that at some point you assess that total body of evidence and you determine when it's plausible enough that a particular group of animals might experience pain that we should start thinking more about their welfare. And so to give you a sense of what plausible enough kind of looks like in the UK context, which is actually what this framework was designed to do, this was commissioned by DEFRA in the UK to better understand invertebrate sentience for legislative purposes. So to understand plausible enough, let's look at 2 groups of animals that have already been assessed using this framework that are invertebrates. The first is cephalopods. Most folks have agreed for, or many folks have agreed for a while, that cephalopods of all the invertebrates are probably the most plausibly sentient. And so they were reviewed under this framework by Birch in 2021 and his colleagues, as well as crabs, the decapod crustaceans. So they found octopuses meet 7 of 8 criteria to a high or very high degree of confidence, which this framework rates as very strong evidence for sentience. Crabs met 5 of 8 to a high or very high degree of confidence, which this framework rates as strong evidence for sentience in that group. And the important thing to know for your context is that on the basis of this report, the UK government included both of these groups in their Animal Welfare and Sentience Act in 2022, which could guarantee future welfare protections for those animals down the line. So this is a legislatively relevant standard of evidence, 5 of 8 or higher. So along with Jonathan Birch's group and led by Lars Chitka, who his PhD student at the time was Matilda Gibbons. She's the first author on this paper. They're from Queen Mary University in London. All of us got together and said, let's review this same framework, but for adult insects. And let's look at 6 different orders that are very well studied for their neurobiology and behavior, because as I'm sure all of you are aware, there are many orders of insects that are not well studied in any context, much less for their neurobiology or behavior. So this is the result of that review. This is a table that matches the one that you just saw for the cephalopods and the decapods. And essentially what I'll call out to you are these 2 blue bars that just appeared, the Diptera and the Blatodia. So those 2 groups meet 6 of 8 criteria to a high or very high degree of confidence at the adult life stage. That's 1 more than the decapods, 1 less than the cephalopods. And all of the areas where you're seeing yellow stars are areas where, in my very casual reading of this literature, new papers have come out that support additional evidence in those areas since this review paper was published in 2022, which looked at a total of 350 papers published across these orders. So what this suggests is that at least in some insects, there is strong evidence for sentience at the adult life stage. I'll also note that in most of the categories where you're seeing a gray box, it says VL, no research found. That stands for very low, no research found. What that means is that we didn't find a single study that could, demonstrate the lack of that criterion in those animals. And you're seeing that in the vast majority of cases, it's just that people haven't even attempted to study the phenomenon in question in insects yet, not that that phenomenon has been found to be lacking. And this kind of shift in the evidence in favor of the plausibility of insect sentience may be matched by some recent surveys and some other movements in the kind of scientific and empirical arena. So this is a study by Zippel et al. That came out last year, and in it they were sort of surveying animal behavior researchers about emotional responses in animals, where again, emotions are subjective experiences. So that would be an equivalent phrase to sentience would be having an emotional response. And so if you look at this graph, essentially dark green is all or nearly all members of this taxon have emotional responses. Light green is most, light yellow is some, and dark yellow is no members. And what we're seeing if we add together the light yellow, the light green, and the green, right, that's folks who are willing to say at least some insects display this phenomenon. At least 67% of animal behavior researchers are ascribing emotions or sentience to at least some insects, which is pretty in accord with what I just showed on the last slide. And we're seeing some other kinds of movements in consensus statements in scientific societies. So the New York Declaration on Animal Consciousness, which was a follow-up in some respects to the 2012 Cambridge Declaration, both of those actually recognize insects as plausibly sentient and deserving of some kind of welfare consideration. Similarly, the Royal Entomological Society made a statement on the ethical treatment of insects last year that acknowledges that there is uncertainty about sentience in insects, but that risk mitigation tools and welfare assessment tools may be valuable to try and help us minimize harm in research and management settings. And this is a version of what we call precautionary reasoning. So the precautionary principle is this ethical idea that when you're uncertain if you may be causing harm, you should act as though you are to avoid causing that harm. And so what that means is that we may want to treat insects as though they may feel pain while we wait for more evidence on sentience to come in and consider how we can avoid unnecessary harms. And so that's going to motivate a lot of what we talk about for the rest of today. So that was a very brief introduction to insect sentience. There's a tremendous amount of literature out there that I think everyone should read. It's fascinating, but kind of this is the summary slide on that. It's far from definitively proven. There's enough evidence for it in some insects to take reasonable precautions to avoid unnecessary harms, and we should be considering welfare while continuing to research proxies for sentience and doing other research we believe is valuable. That's a good course of action for going forward. If you like listening to me talk at this pace, but you want to hear me talk for 2.5 hours at this pace on insect pain and sentience, this is a podcast episode I did a couple of years ago that is still pretty relevant and up to date, and you might enjoy it with the group 80,000 hours. Okay, so we've covered some of that motivational evidence. Now let's talk about what kinds of ways we might go about trying to make collecting efforts more humane. So this part of the talk is going to talk about asking new questions when we go collecting. And a lot of that has to do with this framework of harm and benefit analysis, where we're trying to minimize harms and maximize benefit. And so I'm going to start by talking about the minimize harm part, and then I'm going to talk about the maximize benefit part. So the 3 Rs is a framework that is designed to help think through how to minimize harm to animals broadly when they're used in research projects. And the 3 Rs stand for replace, reduce, and refine. So replace basically means if there are suitable alternative methods other than using live animals, don't use the live animals. But a lot of times, as I'm sure we're all aware, you need live animals to answer your question. And so then you get to reducing. Use the smallest number necessary for the experiment to be valid, right? You don't want to hurt any of your reproducibility measures, the generalizability of your study, the validity of your study, but you want to use the smallest number that you can to achieve highly reproducible, valid, and generalizable results. And after that, you get to a point where you're saying, okay, I'm going to use this number of animals of this type. Now you get to refinement. How can you do anything to try and reduce stress or pain or harm to the animals while they're being used in the procedure? So this framework, the replace, reduce, refine framework, has been promoted by some folks as best practices for other reasons, like just trying to be more thoughtful in conservation practice, where we are sampling a lot of insects from the wild, but we know their numbers are declining. And so maybe we want to start thinking through best practices, not even just for welfare reasons, but also for, conservation and environmental reasons. So this is a framework that aligns really well with a lot of different values. So the first question it's going to ask of us is, can we replace insects with non-insect alternatives? So that would be things like, can we answer our question with previously collected data or individuals? That would be like one of the fundamental kinds of replacement. Could we use indirect molecular and chemical methods, et cetera? Could we use images instead? A lot of those approaches just aren't going to be useful to us if we're not associated with a research laboratory, or we're amateur entomologists or entomologists that are associated with a museum that doesn't have those technologies available. So a lot of times you have reasons why you can't replace, and that's okay. The 3 Rs framework is designed for you to be able to follow the replace decision directly on to reduction. So I think there's this idea for some folks when they think through the 3 Rs that they think, oh, they must replace, even if it's, scientifically bad, or, if there's any possible alternative, even if it's not suitable. And the answer is that's not true, right? You just need to justify why you're using the live animals in your thought process and decision making to make sure that you have a reason why you're using live animals and you can't do something else. And so I think in the vast majority of contexts that you guys are going to be in, if I'm talking to general collectors out there, you're probably likely to be in a case where you're thinking more about reduction and refinement than replacement. I always start my own thinking there, even though I also do a ton of live animal research in my lab, just because I think it's best practice to always be asking myself if I can replace, even if largely I find that I can't. So this brings us to the reduce step. In particular, I want us all to be thinking about bycatch when we think about this particular question, because again, bycatch tends to be that set of our sample that is not nearly as helpful to us as our actual intended sample target species. So when we think about reducing sample size, there's a lot of things we can think about. So I have a little list here about, survey frequency at the site. So can we reduce how often we're surveying? Can we reduce the number of sites that we have? Can we change survey methods from passive to active methods that allow for more targeted sampling? Could we incorporate excluders like what you're seeing over a pitfall trap on the right-hand side that might exclude some large-bodied butterflies, moths, or bees from the trap accidentally, as well as some vertebrates that might find their way in there otherwise? Those are the kinds of things that we can be thinking about for reduction. If you're collecting animals and you already have a lot of that species, you might say, oh, is this actually a group that I can, catch and release? So document that I have them, but not collect another. And another thing that can help with reduction is just coordinating better with others to avoid duplicating our efforts. So if we're collecting in an area, we might not need as many other people collecting in that area with us at the same time, at the same sites, using the same methods to avoid duplicating our efforts. So coordination can be helpful there too. I've also thrown up two guides that we've produced at the Insect Welfare Research Society on power analyses. These are ways that researchers can estimate what kind of sample size they actually need for a particular kind of study design. There's also a great tool out there called G-Power that you can use for the same kind of thing. But if you're interested in kind of a statistical approach to thinking about how many sites you need, how frequently you should survey them, that kind of technique and a priori power analysis can be really helpful, and the guides go through how to do that. Okay, now, in many cases, we can't reduce anymore, right? We've thought through how we can exclude as much bycatch as possible, but we are kind of committed to a particular approach and set of methods. So now it's time to think about refinement. So refinement is this question of, can we reduce the harm to the individuals that are going to be used? So the way that this particular paper that I was talking about, this conservation paper, is framing that, they're thinking mostly about avoiding lethal take or permanent removal entirely, because again, this is for conservation and environmental reasons. So they're talking about things like, can you do, tissue sampling from the animal where you remove a tarsus and just use that? Or could you use capture and release to document the individual? This is employed by some surveying folks here in the US. So I used to chat a little bit with the folks at the California Bumblebee Atlas Project. And as you're all probably aware, bumblebees are large, and you can often actually see many identifying features with just a couple of really high quality photos. And so they actually did have a capture and release program for all of their bumblebees, keeping in mind that, further methodological advances may come beyond just bumblebee research to help folks with other taxa also be able to employ those kinds of less lethal or less permanent removal strategies. But sometimes we do need a lethal method. I've worked a lot with like lasioglossum bees, for example, that you are simply not going to identify, on the wing. And so many cases, we really do need to actually collect these individuals lethally. In those cases, we might be thinking about refinements that allow us to use faster or more humane euthanasia methods. We could consider using anesthetics prior to euthanasia, and more details will be provided on that shortly, because we'll get into the specific methods that the museum mentioned to me folks are using right now in the UK. I just wanted to briefly mention that there are some tools and papers out there that if this is a topic that interests you, you might want to take a look at. So again, Insect Welfare Research Society, we've produced some guidelines for protecting and promoting insect welfare in research settings that might be valuable for folks to take a look at. Just very brief guidelines on refinement and things like that. There's a great website called the 3 R's Principle in Wildlife Research. This is all about what kinds of methods are out there and available that can, replace, reduce, or refine in wildlife research settings. And it does have sections on arachnids, insects, aquatic invertebrates as well, although it is largely dominated by vertebrate methodology. The last thing is this great review paper that came out last year by Levy and Ferrante, The Use and Prospects of Non-Lethal Methods in Entomology, published in the Annual Reviews of Entomology. And that's also a really great paper for just thinking through what some of the non-lethal methods we might have available to us are. And I'm sure it's already out of date with the advent of AI and all the kinds of technological progress we'll be able to make identifying insects with AI tools. Okay, so that's walked us through the minimizing harm section, but let's also talk about this other piece of it, which is maximizing the benefit that we can get from each of our specimens, which I'm sure is something that folks in this group think about a lot already. So when it comes to maximizing benefit, a lot of this has to do with how we keep our samples once we've lethally collected them, or even if it's not a lethal collection, once we've removed some amount of tissue from that individual. And so a lot of this comes down to, for each specimen, considering non-destructive methods, if we are, doing some kind of research study that allow for the reuse of that specimen over and over, storing it safely so it doesn't accidentally get degraded, making sure we keep really good metadata associated with our specimens so that we can deposit them in museums and have them be worthwhile to people for centuries to come. And if we do have a lot of bycatch, we could consider saving and exchanging that bycatch with others who are a little more familiar with that taxonomic group and might be able to make better progress with those samples than we would be able to, instead of just throwing out that bycatch and having it not be particularly useful to anyone at all. We have a paper that's a bit about this. It's admittedly a bit more focused on researchers at academic institutions, but it came out in agricultural and forest entomology on the case for open research in entomology. And one of the things we talk about here is the way that open research can really maximize the benefit we get from animals, including insects. And so if we have high quality data and metadata, if we make that publicly and neatly available to others so that they can reuse it, all of that is going to make it so that these specimens are maximally valuable to others. And so if there are ways that you think you could get more information out there in the world about the specimens you've collected, whether that's, again, depositing them in museums, having a digital database that you contribute to, all of that is a way to help maximize the benefit we get from these animals. Okay, so now I want to get into some of those practical details of refinements for lethal methods, because again, we can ask these new questions about replacement and reduction, but a lot of us are going to get to the point where we are focused more on the refinement piece of the three R's. So when we're thinking about humane collection or humane death, we tend to think of two factors: the duration of pain or distress that the animal might experience, and the intensity of the pain or distress that the animal might experience. And we can try, when we're refining things, to minimize these factors in the following ways. We can use methods that are shorter in duration, causing, a shorter duration of pain or distress. We can use methods that we think are less painful or less distressing. Another kind of way to do that is to render the animals insensate or unconscious prior to the experience of pain or distress, so that the method employed, it doesn't matter whether it would be painful or distressing because the animal is not experiencing anything. And we tend to do that using anesthetics or stunning methods. Importantly, sometimes these considerations can be in tension. So, for example, sometimes a method can be much faster, but we also have reason to suspect it would be much more painful. And so then you have to ask yourself a question about trade-offs of, how much faster is it worth it for it being that much more painful? The other thing that I'll say is that when we talk about humane deaths for animals in research, one thing that folks really stress is ensuring that death actually occurs. And so I know some of us have probably had the experience where you pin up your insects and then you open the collection box a couple of days later and there's a bug and it seems to be wriggling around on the pin. And you thought it was dead, but your ethyl acetate dose wasn't high enough or whatever. And so actually that insect woke up on the pin. So we want to make sure when we're talking about humane death that we have actually really killed the individual using the method intended, and that we're not letting them accidentally starve or desiccate on the pin. So when it comes to understanding intensity of pain or distress in insects, I want to stress that there's a lot of uncertainty around this. The research in this area is very limited and very new. That said, we can at least talk a little bit about how insects respond behaviorally to putatively painful or distressing events, and what kinds of neurobiology they have that might contribute to them sensing particular kinds of putatively painful stimuli. So the sensory cells in your fingertips that I mentioned when I was talking earlier about that example of you touching the hot stove and pulling your hand away, those peripheral sensory cells are called nociceptors. They're cells that specialize, neurons that specialize in sensing damaging stimuli. In humans, they can sense heat, cold, many noxious chemicals, mechanical damage. I'm sure we're all quite familiar with that, having many nociceptors ourselves. But insects, at least some of them that have been studied, we know have nociceptors as well. So we don't know this for every insect because they haven't all been studied, but all the ones that have been studied, we know have nociceptors. And we've done some genetic surveys for the proteins that are embedded in those cells. We've also found good genetic evidence that those proteins are really widespread, giving insects plausibly, of many, many, many orders, the ability to sense heat, cold, chemical, and mechanical damage. Now, we also have some behavioral indicators that these animals are experiencing, or I should say, are sensing some of these stimuli. So they respond behaviorally, right? If you've placed an insect in a vial that is getting heated, you've probably seen them respond as though that situation might be painful or distressing to them. The same thing with, putting them in freezers for cold, many chemicals and mechanical damage. There's actually a whole field of pain research focused on the fruit fly, a biomedical field focused on understanding chronic pain and acute pain in fruit fly larvae and adults as a way to better understand human chronic and acute pain. So we know, and we have a lot of literature on particularly those animals, that there are many behavioral responses to these noxious stimuli, and the insects can at least sense them. We also know that insects seem to experience distress or negative affect when they're placed in situations where their lives are threatened. So that would be like when they're drowning, when they're starving, when there's a predation linked cue. So that could be a looming stimulus or a vibration stimulus, handling, right, which we tend to do when we're using insects in the lab. Those things can lead to elevated stress hormone levels. They can lead to changes in the way the animal responds on a judgment bias test, which is the classic way in the vertebrate literature to assess emotion and affect in vertebrates. So although we don't know a lot, we do know some things, is the point. So when it comes to the current practices that tend to be employed in the UK and what's known or mostly unknown about them, this is the list that was provided to me to prepare for today's talk. So we have immersion in ethanol water or other preservative liquids. We have boiling in water. Typically that's heating up slowly, not direct immersion in the boiling water. We have freezing at minus 20 C, which is your typical freezer temperature. That can be with or without an acclimation period in a refrigerator first at 4 degrees C for a few hours. We have ethyl acetate or chopped cherry laurel leaves, which is not a thing we do in the United States, but apparently leads to cyanide poisoning. It's a little crazy to me that you guys just have cyanide out there on your street corners, but good on you. And then we also have the back of the fridge method where the insects starve or desiccate to death, or sometimes that even occurs not in a fridge where they just starve or desiccate to death at room temperature in a vial. So before I go through each of these methods, I just want to say a few quick caveats here because this is getting recorded. And so I want to make sure for posterity's sake, we're all on the same page. The science here is just emerging. This talk could get out of date really fast. My lab primarily works on stunning and slaughter and anesthesia. That's our major area of focus right now alongside insect neuroscience. And so we are discovering things in our own lab, even hand over fist. So please do try to stay on top of best practice guidance as they emerge. This is a great foundational talk, but of course things are going to be changing over time as better science hopefully happens by many labs around the world. I also want to note that often we're locked into methods for scientific reasons. So like you may just need to use pitfall traps. And so that's just the only method available to you. And I still think it's really valuable to be honest with ourselves and honest with others about the welfare impacts of what we're doing, even when we're locked into a method, so that we can weigh the harms and the benefits of our methods accordingly. Also, if we keep welfare in mind, it may motivate us to experiment with new methods, either ones already developed or ones we want to develop, to see if we need to remain committed to those old and potentially less humane ways, or if there are new possibilities out there for us. So for myself, because I do a lot of lethal and invasive sampling in my lab, I try to take the view of being really honest with myself of the welfare impacts of what I'm doing, while also being really honest with myself about what I need to do for scientific reasons, so that I'm making an all things considered best judgment. The last thing is that I hope this won't frustrate anyone, but I'm not going to just provide you with like a ranked list of what to do. I'm actually going to make very few concrete recommendations. And that's because I think it's better to just provide you, who are really the experts in your situation and your goals and your research and your resources. I just want to provide you with information so you can make your own all things considered best judgment. That is based on, again, your goals, your interests, your abilities, your resources, your tolerance for uncertainty. All those things could factor into what kind of judgment you want to make about what to do. So I'm not going to tell you what to do or be prescriptive. I just want to give you information about what you may be doing. Okay, so let's start with this first method, which is immersion in ethanol water or preservatives. In general, this can take seconds to minutes to hours. That depends on the liquid being used, the concentration used if we're talking about ethanol or preservatives, and the insect's tolerance for being immersed in water and their body size. So all of those things can affect the duration of pain or distress. There was nothing I could find where people are assessing the insect experience in response to drowning. I think those of us who have done that, I have drowned some insects in my day, know that they do seek to escape and respond violently when they're drowning. There is also some research on the forced swim test in Drosophila. So for those of you who aren't familiar, the forced swim test is classically done in like vertebrates like mice who are being studied for depression research, where they essentially put them in a liquid that they can't escape from, in water they can't escape from. And then they just see how long it takes for them to give up on swimming. And that can give you a sense of how depressed the animal is and can also contribute to them being depressed. And so they have validated that that same technique works in insects, suggesting that drowning could have some negative affect experiences, and you can rescue their interest in continuing to try to swim with human antidepressant or anti-anxiety medications. I've also noted in my own experience that insects do seem to respond even more vigorously when I immerse them in preservative liquids versus non-preservative liquids. I don't know why that is. I could imagine that it, has to do with the activation of nociceptors because of those preservative liquids, or perhaps that they increase the rate of osmotic shock. All of that could potentially be pain-inducing or distressing. I don't know. But in my experience, the preservatives do tend to shorten the duration of time to death, even if they increase the appearance of the intensity of the method. Some recommendations that could potentially be made for this method: if you use ethanol or preservatives that you have made ice cold, that you've kept in the freezer beforehand, or very, very cold water, this could reduce the duration as well, because it could lead to much more rapid chilling of the insect. Thermal energy transfer is much faster in liquids like water than in things like air. And so the heat transfer could be very fast compared to in-air cooling and lead to the rapid loss of consciousness. That's especially true if insects are really small or really thin. So if you are able to carry like a freezer out with you into the field, full of ice with your ice cold ethanol in it, that is one method you might be able to use to try and minimize the duration of that kind of distressing period of time. Also, just make sure that you are leaving the insects in until you're very sure they're dead. Again, we want to make sure that we're not kind of causing situations where insects are not quite dead and then suffer for a prolonged period of time as a result of not being quite dead when they're removed from the solution. The next method that was shared with me was boiling water. So I will tell you from experience, I work on boiling of insects for the context that I work on is insects farmed as food and feed, where they can be immersed directly in the boiling water. And that tends to, if you just take a single small-bodied insect and immerse them in boiling water, it tends to result in instantaneous death from my experience. Now, there are some things that can affect that duration question. So one would be a really large-bodied insect like a Madagascar hissing cockroach may not die instantly the way that, a black soldier fly larva would or a cricket would. So that's one thing to keep in mind, right? The more insulation there is for the central nervous system located in the center of the body, the larger the mass of the animal, the more thermal inertia it has, the longer it's going to take for, that heat energy to penetrate, the less instantaneous it's going to be. So generally, smaller bodied insects, immersing them directly in boiling water will work for instantaneous death. Also, just keep in mind how many insects or how much material you'd be immersing, because the more material you are immersing that starts out at room temperature into your boiling water, the more you are going to decrease the temperature of the water and thus decrease the likelihood the animal will die instantly. And even a change by about 20 degrees C in the temperature of the water from boiling to not can change the likelihood of the animal dying instantly from some experience from our lab. When it comes to placing the insect in a jar and then placing that jar in the boiling water, that's obviously going to result in a much slower death. And that seems to be, from what I've heard, a more common practice. Those of you who have done this, and I actually did CT and AX assays this way during my dissertation work, you would know that it can take many minutes, depending on the water temperature, the body size of the insect, and their thermal tolerance. These methods are likely to be very painful insofar as they are experienced. So insects we know have heat-based nociception. They respond with vigorous escape behaviors in global heat assays. It is one of the most consistently used aversive stimuli in aversive learning assays because insects respond so consistently and aversively to heat. So what that means is that we really should try to minimize the duration as much as possible. Ways that we can do that: direct contact with the heated water, if that works for your method, using the hottest water possible, doing one insect at a time to maximize heat transfer. Do not chill the insect in advance, because the cooler the body temperature of the insect, the longer it will take for them to ramp up to their lethal temperature. So, making sure that they have not been chilled in advance will be good. All of those are things that can help you minimize the duration of time. Also, if you are going to be immersing them in a jar, using the smallest possible jar so that you are talking about, again, the most rapid heat transfer through the jar wall, the air, and then into the insect body can also be helpful. Again, do leave the insects in until you're absolutely sure they're dead. If you leave them for 10 seconds when they're immersed in the boiling water, they will definitely be dead for these small-bodied insects. But if you're not doing a direct immersion, you'll want to leave them for long enough that you are really sure they have finished. Typically insects in this case, they'll sort of fall over and convulse. This is a seizure behavior that they do before the paralysis that's the kind of official onset marker of death. Leave them for several seconds after that onset of paralysis to ensure that they have actually died. Moving on to another thermal method, which is freezing at minus 20 degrees C. We know that at least some insects can sense noxious cold. This has been shown again in Drosophila, which is our major research model for biomedical research on noxious stimuli and pain in humans. Many insects thus seek to escape noxiously cold environments, or they seek to heat themselves up if they have that ability when they're in cold environments. So bumblebees, for example, if you put them in the freezer, they'll kind of shiver their flight muscles internally to try and heat themselves up and delay getting cold. One thing to note is that the periphery of the animal and the central nervous system, they may cool at essentially the same time, but in many insects, they won't, right? So thermal energy transfer, just like I was talking about, it takes some time to penetrate from the exterior surface of the animal to the interior. And freezing is happening in air. Air is a much slower medium for heat transfer than things like liquids like water. So what that means is essentially that the periphery might cool first, but the central nervous system may still be active during that time. The insect may still be sensate. And it can be hard to know whether that's happening because of what we call this locomotor confound. So if you've ever been really cold outside, you'll know that your fingers, right, your periphery, they start to not work so well. You can't really move them the way you're supposed to. And that happens to insects when they get cold. Their periphery, as it chills, becomes immobilized and they can't move, even if they still technically are having a central nervous system response. So it's important to note that you should definitely consider, especially for larger bodied insects, leaving them for longer in the freezer to ensure that you're not just seeing an immobilized insect, you're seeing a genuinely chilled insect where chill coma has been induced all the way down to the CNS. One other thing to note is that cold itself is not considered a valid analgesic. It has no analgesic or pain relieving properties. However, it can induce unconsciousness, right? So once chill coma has been achieved, unconsciousness has been achieved in that animal. The unfortunate thing about using cold as an anesthetic method, though, to generate unconsciousness is that, distress can be experienced by the animal before that. And like I just mentioned, you can't actually ever assess the animal has genuinely been anesthetized, which we call anesthetic depth. You can't assess anesthetic depth because of that locomotor confound. So when it comes to freezing, the invertebrate veterinarians at BIAZA, which is the British and Irish Association of Zoos and Aquaria, they have a terrestrial invertebrate working group that makes recommendations for the zoos and aquaria on how to treat their terrestrial invertebrates. They don't currently recommend freezing as the way to kill insects, but recognizing that this method may sometimes need to be used by staff, their recommendation is to place the insect in a fridge for 4 hours, which is thought to kind of more gently acclimate them to cooler temperatures, and then move them to the coldest available freezer immediately after that. The idea there is that then because the insect is already at a pretty cold temperature, by moving them to the freezer, you minimize the amount of time that they will spend chilling from that cold temperature to the coldest temperature. The other thing that you can do is you can actually pre-chill tubes that are in your freezer and then transfer the insect to that pre-frozen tube. And that will again, through direct conductive heat transfer with the insect touching the tube, can actually result in even faster time to death. If these options are available to use, using liquid nitrogen or dry ice can result in really rapid freezing, and that can even potentially make death instant if there's direct contact between the LN2 or the dry ice and the small-bodied insects, but I don't know how easy those things are to get in the UK. They're reasonably easy to get here in the US. When it comes to confirming death with the freezing at minus 20 C, as some of you may be aware, it can also be really hard to confirm death there. So leaving them for longer is generally better. I always leave the insects in my freezer for at least 24 hours if I'm doing a freezing method to try and ensure that they have died. Longer than that if they're like embedded in a substrate or I have some reason to think that they might be able to maintain a higher body temperature for longer. Next method up is ethyl acetate. So ethyl acetate is sometimes referred to as an anesthetic for insects because it can induce unconsciousness. I couldn't find any major veterinary sources reporting this actually as an anesthetic recognized by their community for insects or for other animals, but entomological sources do refer to ethyl acetate as an anesthetic. The reason for the difference in the use of this term by veterinarians versus entomologists might be because these communities use the word differently because they have different goals. So many veterinarians are thinking of an anesthetic as something that isn't causing significant distress on knockdown, where you can assess anesthetic depth, where you're not causing extraordinary toxicity to the animal during the anesthetic unconsciousness period, whereas many entomologists who are using the term anesthetic really just mean, is the animal technically unconscious or immobile? Not caring as much about the question of distress on knockdown and things like that. So I just want to note that I'm not going to try to resolve that debate for you here today. I think more guidance by veterinarians on ethyl acetate specifically would be valuable, but it's worth noting that there's a different compound that sounds similar but is different, diethyl ether, that they do explicitly not recommend for use with insects at this time. So what we know is that some insects do seem to respond aversively to the agent when they're placed in the jar. This especially can happen at higher concentrations. It tends to only last for a few seconds before knockdown, and that's true for most insects, although those that are really hypoxia tolerant sometimes actually are able to kind of last for much longer in the ethyl acetate kill jar. In general, you might recommend using higher concentrations and ensuring the kill jar remains full throughout the day, just so that you're ensuring that you're minimizing the duration of time that the animal is exposed to the ethyl acetate during knockdown. Really high concentrations do seem to result in pretty efficient knockdown for most insects. The other challenge with ethyl acetate that many of you are probably aware of and that I've had happen myself is that it is, again, basically impossible to tell when the insects are dead. The insects often wake up on the pin afterwards because it appears as though they have, stopped breathing and everything else, and yet it actually just turns out that they were close to death but wake back up. So using a secondary method of death when you can, if you're going to use ethyl acetate, along with keeping your kill jar really stocked with high concentrations of ethyl acetate, those two things can help ensure that death has occurred. If you just take the animal in the kill jar, put it directly into the freezer while they're still unconscious, that might be a great way to ensure that death has actually occurred and prevent the animals from waking up on the pin. The other thing is this can actually wait until after transport, assuming that your kill jar is always well stocked, because in theory, assuming your kill jar is well stocked, the animal will be unconscious from the moment of knockdown all the way through until you get back from wherever you've transported them. So chopped cherry laurel leaves can apparently cause cyanide poisoning. There's some chemical reaction that occurs when you chop the leaves, mixes some chemicals together, and can release cyanide gas. And so cyanide poisoning, the way that seems to work is that it causes internal suffocation. This could be very slow in insects, actually, as a result of their very low metabolic rates. So it may be that they lose consciousness quickly, but then that death is actually quite slow. Because the loss of consciousness and the loss of motor function, that tends to occur faster than the actual death does. The major challenge that I've seen reported with this method is getting the dosing right. The leaves lose their potency over time pretty quickly, and it's the freshly chopped leaves that give high doses. So it's really hard also to be standardized in your dosing with this method because, the leaves have variable amounts of the chemicals in them. When you cut them, it creates variable amounts of release. And so just getting high doses is really, really hard to do. Plausibly, again, using higher doses probably the best practice, and restocking your jar very frequently to keep those doses high is probably really important. You may want to have several jars that you can switch between for both the ethyl acetate and the cherry laurel, so that you're always using a jar that is really freshly stocked. And then you're able to kind of restock old jars while you're switching to your new jar. A last, challenge with this one, again, very similar to ethyl acetate, basically impossible to tell when the insects are dead. They regularly wake up on the pin. You should use some secondary method. The potency concern is more challenging and harder to manage with the cherry laurel leaves than it is with the ethyl acetate because of the aforementioned dosing issues. So it may actually be that you want to use that secondary method immediately after the animal appears unconscious instead of transporting them back to your home to freeze them. So bringing a freezer with you into the field, that might be a great way to go if you're able to, just so that the animal isn't waking up during that transport period as the dose of the leaves wanes. When it comes to this last method, which is sort of like the starvation or desiccation, either in the fridge or outside the fridge, this is by far the slowest of the methods described. It can take days or months. This is especially true for insects that have a lot of lipids stored that are not actively using them. And again, it can be really hard to know when the insect is definitively dead in these cases, especially, if they're going to be pupa that don't move, for example. It's very hard to know. Like the science is really not clear about what insects that are like in hibernation or that are pupa might experience at all about really anything. We have much less awareness or information about the neurobiology of pupal insects because they're hard to study because their neurobiology is changing. We also just have much less understanding of, their their responses because they're often immobile and unable to behave. Um, so that said, many of them are going to have adult-like brains if they're kind of at the tail end of pupation, and some of them, even as you're aware, retain a degree of motor control, like yellow mealworm pupa that can do that flicking response with their abdomen. If you need to use this method over others, because I appreciate that there are reasons why people are using this method to kind of reduce the lipid content of the sample, anything that you can do that can hasten the duration. So instead of letting the insect desiccate at room temperature, pack them into desiccant crystals, right? Something like a dry rite or those little, packets that you get with clothes. That seems preferable. I have a really high degree of uncertainty about that, but in general, right now, I tend to take the approach that minimizing duration is a good thing with most of these methods because we know so little about the degree of pain or distress, but we do know we can shorten durations. And so I would favor myself right now things that hasten the duration accordingly. If you're looking for more guidance on this, you might like this publication by Roy and Vidal-Naquat in 2022 in the Canadian Veterinary Journal. They were going through and making some recommendations on the euthanasia of whole honeybee colonies. Obviously, there are some unique constraints to trying to do euthanasia of an entire colony at the same time. The column that's most relevant to our discussion here today is the quickness column, where, again, they're trying to talk about minimizing duration as a measure of humaneness, but they also do talk about, what it would look like to consider cost, complexity, operator risk, and many other disadvantages as well. So you might consider taking a look at that publication. The other thing that we can talk about for refinements is using anesthetic agents prior to methods that we don't think can be made instant. Um, that would be, again, things like where you're, heating up your insect in a jar that gets immersed in boiling water. That's never going to be instant. We expect it may result in pain or distress insofar as insects experience that. And so using an anesthetic option prior to using that method might really be the only option for making the method humane. So here I want to say I'm not an anesthesiologist. I'm using guidance produced by veterinarians who are, trained in animal anesthesia, including many of them specializing on invertebrates. And majorly, I've taken my information from this Anesthesia and Analgesia in Laboratory Animals book by Dyson et al. There's a whole chapter in it on insects. There's also some chapters on decapods and things like that if you're interested in aquatic animal guidance. So preferred anesthetics don't cause pain or distress on knockdown, and they allow us to assess that the nervous system is actually out. So we're not just seeing a locomotor confound of depressed motor function as a result of it being cold at the periphery. We're seeing actual central nervous system is knocked out. So cold and hypothermia, as I've already mentioned, doesn't meet those criteria. We can't analyze anesthetic depth, and we think it could cause distress on knockdown. Carbon dioxide is something that veterinarians have discussed works for insects. It's considered the less progressive but still effective option. The reason why it's considered less progressive is because it can cause hyperexcitability at really high doses, which is a potential indicator of distress in animals. One thing you could potentially do to kind of reduce the likelihood of this happening is just begin, with a small trickle of CO2 at a low dose and then raise the dose slowly over time to kind of minimize the likelihood of hyperexcitability. And kind of use the minimal dose required to get the animal to actually be knocked out. The preferred methods for terrestrial invertebrates are halogenated ethers like isoflurane and sevoflurane. However, those can only be obtained with veterinary assistance, particularly in the UK. That's because they do also work on vertebrate animals like us and have some safety concerns. And so they're probably not going to be an option for most people that are not themselves veterinarians or very good friends of a veterinarian who wants to help them improve their practice in this way. So carbon dioxide is probably the most likely to be available to folks on this call. When it comes to aquatic invertebrate species, the two things that are available are MS-222, which is commonly used for decapods, and again, actually carbon dioxide, but bubbled through the water. There's some discussion of that in Cooper 2011, where John Cooper founded the Veterinary Invertebrate Society and was publishing on this all the way back then. In terms of getting carbon dioxide for the field, there are some options that are pretty readily available, again, depending on your context. So one thing would be that many folks need to carbonate kegs, soda bottles. They want to handheld carbonate their kombucha. And so there are actually these carbonation devices that people sell for soda bottles, for kegs, et cetera. And so I think the best one is the one you can see in the middle here, which was called a keg charger or a handheld kombucha carbonator. Those are the two terms people used for it. And you can see it has just a little nozzle and a little canister that gets filled with CO2. And essentially, it's just going to allow you to press this little like trigger, and then it's going to shoot CO2 out the nozzle into whatever collecting tube that you have if you just stick this little needle through that collecting tube. So that is a method that might work for some folks that are interested in kind of being able to choose the flow rate of their CO2. And you can just purchase big packs of these CO2 canisters if you don't want to have to refill them. Another thing is that dry ice is just a lot of CO2. So insofar as you might think, oh, I want to use this method, you could either use the CO2 itself for the euthanasia method, or you could potentially use it to just create a very, if you let it sublimate, a very CO2 enriched environment. The last thing I found was this soda maker method that you can find freely available online. It's a study where they essentially make a little carbon dioxide creator in a little soda machine as a way to try and help with non-lethal identification of wild insects by anesthetizing them in the field. So readily available with a variety of different methods. If you want to learn more about understanding potential pain or stress in insects, I've popped some publications that are some of either mine or some of my favorite papers on Drosophila up here on the screen. Here are some more of my favorite papers on potential pain or stress or indicators of physiological stress. And if you have any questions on the stuff that I've shared so far, or you want access to some of the resources like that book chapter that I was talking about, if you want to see more of that information or get more specific guidance, you can send us an email at admin@insectwelfare.com, and we'd be happy to share more of those resources and guidance with you on a case-by-case basis. These are more resources specifically focused on anesthesia and euthanasia in insects. So a bunch of these are produced by veterinarians, including the first four resources, and then the bottom two. One is a test of different Drosophila anesthetics, and on the left is a preprint of mine, where we actually go through and look at a couple of different orders of insects, specific species that are models, and what practices people are currently employing in lab settings with a variety of different anesthetic agents being employed. Okay, this brings us to this rapid fire last little bit before we get to Q&A. If you're interested in kind of the discourse on the ethics of collecting insects, all of these are papers that you might find really valuable. Some of them talk about these automated or non-lethal insect monitoring methods that have just come out. So I'd highly recommend taking a look at some of those publications, like the Insect Conservation and Diversity one in the bottom left. Also, there's been some papers, including one of my own, where we just discuss what the general conversation is in the community about lethal insect monitoring, like what you can see in the upper left. Here are a few more papers on that exact same conversation by myself and colleagues. And then there's some tools for ethical insect use and research that I already mentioned to you all. Some groups that are really interested in the ethical treatment of insects, like the Veterinary Invertebrate Society, which is in the UK, the Royal Entomological Society, obviously in the UK, and us at the Insect Welfare Research Society. We also have a research library you can search if you're interested in learning more about insect welfare and ethical treatment of invertebrates. We have a newsletter we sent out once a month that has a lot of these kind of research articles that are coming out about non-lethal methods featured in it. And we also do host a seminar series where some of these topics come up. Okay, so in conclusion, if you fell asleep and you just woke up now, what you missed, it's plausible some insects may be sentient, and so we should consider avoiding harms that aren't necessary, right? Some harms are necessary in our research. We should avoid causing unnecessary harms to them. Using the 3Rs framework can help us think through ways that we can minimize potential harm and maximize potential benefit. That means replacing and using live insects only when they're necessary, reducing the number that we use, especially if we can do that with bycatch, refining our practices to promote faster death, more humane death, using anesthetics where possible, staying on top of our methods because we know that knowledge in this field is shifting. Responsible specimen stewardship, right? That encourages reuse of our specimens with the best available metadata to maximize the benefit of this resource to the community. With that, I'd like to thank all the folks who had conversations with me here today, helped organize this workshop, including especially Gary, Ian, and Leanna, and the many, many authors of the works that are cited herein who have done the bulk of the work to make this talk possible for you today. And with that, I think we'll switch to questions.